That is a lack of enthusiasm right there. All right, well, nice to see you guys. Today is the 10th, and it's Dress Up Like an Old Person Day, which, no. Oh, okay, gotcha. Oh, okay, all right, got a couple. Two, I just dressed as myself, because there you go. I'm old. Today is the 10th. What's the dress-up day tomorrow? Mums? Oh, yeah, because it's Pep Rally Homecoming time. I always think of that. In my brain, I always divert to that being Friday, but that's Thursday, because we share a stadium with the school that tried to steal it all from us. So today's the 10th. Next class is pre-lab. So this is Wednesday. I'll see you guys on Friday. We'll pre-lab and we'll kind of like finish up chapter 5. Chapter 5 is all the like membrane transport stuff. And then after that, we have lab in chapter 4. Chapter 4 is like all the organelles, nucleus, mitochondria, chloroplasts, that kind of thing. So chapter 5 is all of this, and then chapter 4 is like all the stuff on the inside. Next time we have something big, I know I told you guys this last time, but the next time like something is due is right here for my odd day classes, you guys. We've got a quiz coming up on the 18th. You'll probably be happy to know it's a little bit more multiple choice heavy, 7 multiple choice, 2 short response compared to our first one. And then after that, the labs are due. Then we have a day, and then we have a test. So this is where I was kind of talking about how biology kind of like compresses and then sort of relaxes. So this is where it gets kind of like crunch time a little bit right there. Yeah, I think that's it for announcements. Do you guys have any questions about announcement stuff? For your labs, they're always due at the beginning of class. So on the due date, if you're in an odd class like you guys, then they're due at the beginning of the 22nd. If you're my even day class, 6th period, and you're watching the video, then it'd be due on even day at the beginning of class. And then each calendar day after that, it's -10, which is a big deal because they count as summatives. So if I turn it in like 3 calendar days late, 4 calendar days late, and you're like, how late can I turn it in? I mean, at some point, there aren't any points left. So if it's like 8 days late, you're at -80 already. So unless you have a perfect paper, you can't get better than like a 20. So make sure that you stay on top of those. And I think that's it for announcement stuff. So yeah, anything we need to know about homecoming stuff? Saturday's the dance, right? Today's nothing. Tomorrow is the game and homecoming stuff. All right, cool. Good luck to y'all that are doing stuff in the pep rally tomorrow. All right, so first off, we had simple diffusion. What determines what way things move with simple diffusion again? They move from this concentration to this concentration. It was from high to low, which apparently is represented with this. So simple diffusion is things moving from high to low concentration. And then if I want to kind of help this thing out, then I can do some helped diffusion. That is facilitated diffusion. And the simplest way I do that is with a channel. Stuff would still move from like high concentration to low concentration, like that. Let me switch over to this camera. Cool. And then what was it called if like water went through here? If water went through a channel? Yeah, they call that osmosis. So osmosis, if water happens to be the thing that's like going in or going out. There's another type of facilitated diffusion, another type of channel. What could I do if I want this channel to not be open all the time? What can I install on it? Yeah, a gate. So I can have a gated channel. So we saw that one last time. Even though it might bother you, you might be like, hey, to open the gate, doesn't that require energy? Sometimes yes, sometimes no. Sometimes like pressure opens the gate, and that's why I have a sensation. Sometimes in my eyes, light opens the gates, and that's why if I go like this, then like I see all the colors. Sometimes sound, is what opens the gate. So it's not always like the cell has to do the opening. Sometimes I'm like literally smashing open the gates, and that's how I feel pressure. One thing that's kind of interesting that came up in a different class was one thing that's weird about your brain is like all nerve cells, I mean, there's a few different types, but nerve cells all work essentially the same way. And so like the nerve cells that I hear with or see with or I store memories with or I learn how to write or read or something like that, many of them are identical brain cells. It's just where they're located in my brain determines what they do. So when the brain cells in the very back of my brain, we didn't talk about this in this class, right? Yeah. So the brain cells in the very back of my brain, whenever those fire, like I generate the illusion of light. So anytime these guys fire, I'm going to see light. My right ear has a spot over here. My left ear has a spot over here on my brain. Whenever those light up, then I am going to hear a sound. But what's strange is like the nerve cells are identical, kind of like on the periodic table. If I have carbon, what makes carbon carbon is it has 6 protons. If I add a 7th proton, all of a sudden it's not like hard carbon anymore. Now it's like a gas, it's nitrogen. But I just added another proton. It seems weird. It's kind of like if I have 6 grains of sand, and it's like solid and it's sand, but if all of a sudden I add like a 7th grain of just more sand, it's not sandy anymore, it's like a gas. Isn't that strange? Wouldn't you think that like the fundamental things that make up like an oxygen, a carbon, gold would be different? It wouldn't be like, oh yeah, how many protons you got? Well, now it's like this metal called gold. Oh, you got a different number of the same crap? Now it's this totally different element. It's strange. So again, one of the puzzles of neuroscience is like, I've got this nerve cell. I've got this identical nerve cell. If this one's located back here, then if it fires, my brain says light. If this one fires, my brain says sound. So we can kind of prove this really quick. If I close my eyes and I look, I think it's bright enough. Actually, I'll do this. So watch me do this first, otherwise you won't be able to see it. So if I close my eyes and like look towards one of the lights, and then I kind of smash my eye on the nose side, not on the outside, but on the nose side. I'm just kind of like pushing on my eye, nothing crazy. Then if I go like that, as you guys walk in, I'm like, if I go like this, tell me if you see like a splash of color. Let's try that. You're not going to look like an idiot, like 6% of you guys are already dressed up like old people. Like it's not going to get any worse. Okay, so how many of y'all, when I do this, you see like on the periphery, like right here in the corner, I kind of see like a little splash of color. Like I see a little halo of blue. If not, you're not pushing hard enough. So if I do that, then I see a splash of color. Okay, was that color really there? No. What did I see then? I saw pressure. My eye has a nerve cell, or nerve cells. My eye has the optic nerve cells that go back to, let's see, that go back to this part of my brain. Normally, the only thing that makes them send a message to that part of my brain is light. Light opens up these gated channels. And if they open up, I see light. Technically, if you happen to be watching this like later on in life, it's sort of the reverse in vision. They're open and then seeing the light closes them, but same idea. I open and close the gate based off of light. Well now, if I push on my eye like that, what happens is back here, I smash my optic nerve. So it sends a message to my brain, and my brain goes, blue? And it just generates blue. But blue's not really there. I saw pressure, and it was blue. Right? I have temperature receptors in my mouth. If I put an ice cube in my mouth, then some of these gates open and close. It sends a message to my brain, and I'm like, that ice cube is cold. But I can also take some room temperature 5 gum. I know this is a notepad. Pretend I have some room temperature 5 gum, and it's like super extra minty mint wintergreen. And I take a couple pieces, I chew it up, and I like kind of smear it across the front of my teeth, and I take a big deep breath. I'm just like, don't I get like brain freeze almost? Like, doesn't it hurt my teeth that's so cold? Are you guys with me on this? Is it actually cold? No, it's room temperature gum. In fact, it's warming up to my body temperature. But I opened up gates that go to my brain that sense cold, and so my brain's like, feel this. And you're like, oh my gosh, that hurts bad. It's so cold. But it's not. It's just that part of my brain lights up. So I find gated channels interesting because that's how I sense the world around me. I might be sitting in my room, it's 2 in the morning, dead quiet. I wake up for some reason, and then out of nowhere, in like my right ear, and it just kind of fades. Tinnitus. How many of y'all have experienced this? There's just this random ringing in your ear. One of the reasons you know it's random is because it's in like one ear. Once you determine it's not a mosquito or whatever. All right, is that sound really there in your room? Nope. You listen to music too loud, or you work road construction, probably for you guys, the first one. You listen to music too loud, so you damage those nerves. And so every once in a while, they just misfire. And whenever that nerve cell fires, it plays a pitch of 1,200 hertz, or whatever that tuning fork sound is. And so my brain's like, oh, this nerve's firing, hear this. Bing. But it's not there. You just open those doors on accident. So anyway, that's why I find gated channels interesting. They explain like why cold gum isn't actually cold, but it tastes cold, why hot things like peppers taste hot even though they're not, why my ears ring, why I can see pressure, all kinds of stuff. It really is all in my head. All right, cool. So if I have diffusion or osmosis in the name, it's always going to be passive transport. So we had these, and this was always, passive transport. Let me just smear that around. There we go. So this was always passive transport. And then our first active transport that we saw was a pump. Why was it again that I would ever use active transport when I could just use passive transport without spending any energy? Yeah. Osmosis and diffusion is always going from high to low. If I want to move stuff from low concentration to high concentration, I need a pump. So if I really like these dots and I want more, even though there's only a couple versus many, if I want to go against the concentration gradient, we say, then I need a pump. All right, cool. We saw one other active transport thing here. That was the one where I was kicking babies around, right? So that was co-transport and counter-transport. What was the difference between co and counter-transport? Like what made co-transport co? Or yeah, the direction they're moving. If they're both moving in or both moving out, they're both going the same way, it's kind of like cooperate together. They're going in the same direction. Counter-transport would mean one of them's going one way and one's going the opposite way. That makes total sense to me. One country launches missiles, that's a strike, and then somebody else would like counter-strike. If I'm in a debate, I make a point, and then you make like a counterpoint. So counter is opposite, co is like together, so that makes sense. And then in this class, I think, did we have one more? Did we do cell identification, like glycoproteins, glycolipids? Okay. So we'll add that one back on in just a second. So that catches us up to where we were. Now we're going to add on some new stuff. Everything so far has usually been like moving molecules in and out like one at a time, two at a time, three at a time, sort of like single file. Almost like if letting you in this room, this door was a whole lot more narrow, you guys could basically only get in like one or two at a time. As opposed to the front of the building, we've got like 10 doors, so I could probably have like 20 students coming in at a time in the front door or the doors by the gym because I have like so many doors. So now what we want to do is look at what we call bulk transports. So bulk transports. That's exactly what it sounds like. If I've got really, really large objects, or I'm just going to bring in like thousands or millions of small objects at a time, then that's bulk transport. All right, so this picture up here, anybody want to take a guess as to what that is? It's kind of like those psychology drawings, so you might see something, but it's really not. Kind of like looking at the clouds and you're like, I see a falcon. I'll tell you what I see. I see a bird with a giant beak, like a toucan, sitting on a branch. He's looking this way. That's his giant toucan beak. He's sort of like this, with like his feathers there, and then he's right there. That's not what it really is, though. That yellow thing, this is a digital image, so this is from an electron microscope. Electron microscopes are digital, so just like anything digital, I can change the colors if I want. So these are not the original colors. So this yellow thing is actually a white blood cell, and it's gobbling up a bunch of E. coli. In real life, white blood cells are not yellow, otherwise the name would be stupid. And E. coli are not bright orange. Otherwise, you'd look at raw meat or whatever, and it'd be like covered with this orange film or whatever. Some bacteria do have colors, though. Oddly enough, the bacteria that likes to grow on soap, which is weird, because soap is what I put on my body to get rid of bacteria. Sometimes those are kind of yellowy, orangey, or pink. So if you're ever like in a shower, and it's not because it's like a streak from like the metal, like the drain or whatever, it's just over in the corner, there's some pink. That means that there's so much bacteria growing in the corner of your shower that you can see it. You can see the pink. So you can see the orange. You can see the kind of yellow colors, like that. All right, so this is a type of bulk transport. I've got endocytosis, and then further down, because I've got 3 different types of endocytosis. Further down, I've also got exocytosis. Exocytosis. So endocytosis, we've got 3 different types. Exocytosis, we've just got 1. Crap leaves the cell. All right, so some of you guys are in a health science pathway, so you probably took medical terminology. Usually that's like a 9th, 10th grade class, something like that, pretty early on. Does anybody know what osis means? Medical terminology. There's a lot of osises in medicine. You got osis, osteoporosis, phagocytosis. It's like a state of or a condition of. That's why a lot of diseases are in osis. Osteoporosis is a condition where my osteo bones are porous. So endocytosis, osis is like a state of or a condition of or like a process. What is cyte? C-Y-T. Cell. There's all kinds of things with cyte, like cytosol, the cytosol, the cell solution, cytoplasm, that's the solution plus all the crap inside. So it's a condition of cell, and then what do you think endo means if it's the opposite of exo? Yeah, enter, go inside. So this is a process of or a condition of stuff entering the cell. So this guy is bringing something inside the cell. Well, they break it down into 3 different types. There's phagocytosis. Phago means to eat. There are even white blood cells called phagocytes because they are good at eating things. There are white blood cells called macrophages. Macro means big because they're big eaters. So phagocytosis is when the stuff that I'm bringing in is mostly solid. And then I've got another type, penocytosis. Well, if phago is cell eating, what would you guess peno is? Yeah, drinking. So now, if the stuff I'm bringing in is mostly liquid, we call it penocytosis. There's always a gray area on certain things where it might be like, would you call this eating or drinking? Just like with real foods. Like everybody would agree that I don't drink pizza. Everybody would agree that I don't eat apple juice. But you start getting into some weird things where you might be like, would I say that I'm eating ranch dressing or drinking ranch dressing or something like that? I'd be like, I don't know, but you're gross. So there can be some gray areas. And then the last one is called, this is my favorite. I'm sure it'll be your favorite too. Is this your favorite type of endocytosis? Receptor-mediated endocytosis. Known as RME on the street. So receptor-mediated endocytosis. Receptor-mediated endocytosis. Bless you. Receptor-mediated endocytosis. Spelling doesn't count on the AP Bio test. In your notes, these things are spelled out also, so even if you like flip-flop some vowels or something like that, no worries. Receptor-mediated endocytosis. So now what I want to do is show you some examples of these up here on the screen. So phagocytosis is the one that we were just looking at. I've got a white blood cell. It rolls up on some E. coli bacteria, so it starts gobbling them up. That's mostly like eating something kind of solid. So we could say that is endocytosis. More specifically, it's cell eating, phagocytosis. Here is a cell. I'm zoomed in on a cell. So this is the bottom of the cell membrane, and it goes like up there off the screen above. And do you see how it's just kind of like pinching inward like that? It's almost like you guys can blow bubbles. So let's say I've got some gum and I blow a bubble. Have you ever done a reverse bubble where you like take the gum and you put it on the front of your teeth and then you like breathe in, you're like, that's a bubble on the inside? So you guys might sometimes wonder, it's like, hey, Cotton, like the internet came out when I was in college. When I was a freshman in college, if you said, where do I get internet? I'd be like, what are you talking about, crazy person? When I was really little, I'll tell you this a few times throughout the year, but just gives you an idea of how far tech has gone. We didn't have hard drives. So when you turned off your computer, it was gone. When you turn it on the next time, it was like every other time you turned on your computer because it was brand new, because there were no hard drives. If you wanted to save something, you had to save it to this little like giant floppy disk, and they held 0.14 megabytes. So if you wanted to play a game might be like 20 floppies. So you're cruising along playing something stupid like Zaxxon, and then all of a sudden it freezes, and you're like, time to load up the next one. So to put that in perspective, your notes, the file for your notes is several megabytes. So that would have taken hundreds of floppy disks back in the day. Like just the notes that you have. Not all the notes that I'll ever give you, but like the notes that you have in your hands. One of those images would take up like multiple things. So my point is if I'm looking at some of these things as they change over time, stuff gets pretty advanced. If we're watching these things like kind of bring something in like this, it's impressive that we can see some of this stuff under a microscope now. But essentially, I kind of bring this in sort of like if I'm, instead of blowing a bubble, I'm like bringing in a bubble. This is the stuff that we did before the internet. You're like, what did you guys do when you had time? You're like, I don't know. Learn to gleek. You know what gleeking is? Kind of hold your tongue a certain way. And if you can like smash the salivary glands under your tongue, like this stream of spit comes out, but it's not foamy spit, it's like just pure spit. So that's what I tried to figure out how to do in 3rd grade. I never succeeded. Sometimes you do it on accident if you're eating a Jolly Rancher. So that's what we did. That's why we are the way that we are. So let's say that you're doing like an internal bubble. That is what pinocytosis looks like. So I've got this cell membrane, and let's say that I have something that's pretty big like a bacteria. So let's say that this guy right here is my bacteria. He's sad because he's getting ready to die. And so now, this cell membrane just sort of like pinches inward, kind of like an internal bubblegum bubble, like that. And now he ends up getting kind of like encapsulated. So now what it would look like is he's sort of in here like this. And he's now surrounded by like this bubble of phospholipid bilayer. Like that. And then when we get into chapter 4 stuff, we'll learn how lysosomes help to like destroy that guy. So if I'm bringing in something mostly solid, that's phagocytosis. Sometimes, though, I just sort of take like a little cellular drink. So let's say I've got a bunch of different particles. I've got chemicals represented by a green pen, a black pen, a couple of different blues, stuff like that. All right, cool. So now, let's say that I just need to get some of these dots out here. Pretend that I really like the green dots. That's what I want. Well, I can just like pinch inward like this, and now I'll just have like a little bubble full of green dots. I also have black dots, blue dots, aqua dots, and stuff like that. But what I really want are the green ones. But I'm going to get whatever's out there. So this kind of pinches inward. Endocytosis. And now whatever is right outside, that's what I'm going to get. So it's a little bit like today. Today's Wednesday, so it's popcorn day. And 10 o'clock, I bet I can't smell it yet. No, not yet. But eventually, by the end of this class, if I open up the door, there's like popcorn particles floating around out there. There's popcorn smell. So imagine this room is a cell, and I just kind of go like this. I get popcorn molecules, but don't I also get like carbon dioxide and oxygen and nitrogen and everything else in the air? Yeah. So it's fast, but it's not that accurate. I'm getting tons of dots, and some of them I want, and some of them I don't. So now we've got kind of a compromise. The next one is RME, receptor-mediated endocytosis. With receptor-mediated endocytosis, here's what I do. So this one, I guess you could say, is phago. This one is peno. And then RME. I'll move this guy over a little bit. So RME. Let's say that there are a bunch of blue triangles. So some chemical with a certain shape. And I want those blue triangles. Now, there's also all kinds of other crap that I don't care about. There's also these other molecules floating around out there. But all I want are the blue triangles. If I get some of the other stuff, fine, but I want as many blue triangles as I can get. Kind of like when I open the door, pretend that I'm wanting to get as much popcorn smell in here as possible. That's cool, but again, I'm also going to get carbon dioxide and oxygen and nitrogen and all the other stuff in the hallway. All right, so what I can do is this. If my body wants blue triangles, then I'll make receptors, proteins that have a certain shape. And I'll just manufacture those receptors in my cell. We'll see how we manufacture them in Unit 6. But I take those receptors and I like ship them up to the cell membrane. So now it looks like this. They kind of look like a elongated capital Y. So I take some of these like individual proteins that have a very specific shape and I ship them to the top of the cell membrane. What I'm wanting them to do is receive blue triangles, so I call them receptors. Receive, receptor. So it's not an enzyme, it's a receptor. Here's the difference. if I've got an enzyme and a substrate goes into an enzyme, I'm going to chemically react and like rip that apart. Sometimes, though, I just want a chemical to fit in there, and that's a way that they communicate. So receptors don't like break down the thing that they touch. They just go together, but everything remains unchanged. Like a real lock and a real key, I can use that lock and key. They go together, but I don't like break any chemical bonds in the lock or any chemical bonds in the key. They're unchanged. So what will happen is I want these blue triangles. I'll manufacture receptors that fit those blue triangles. I'll ship them up to the top. What's going to happen when that blue triangle bumps into that receptor, do you think? Yeah, they'll kind of stick together. They bond. And so now I'm kind of grabbing these blue triangles. Now when I pinch inward, just like these two, aren't I more likely to get more blue triangles? I am, so it's more accurate. I'll give you an analogy in just a second. So sometimes I just pause. If I see tons and tons of people like pencils moving and stuff, I'll just kind of pause and shut up for a second and let you get caught up. So with RME, I make receptors. I ship them to the surface of the cell for the stuff I want to grab. I let them sit on the surface of the cell for a little bit, and then it just kind of like pinches inward just like these guys do. But now I've got a better shot of getting more of those blue triangles. Think of it like this. Pretend that I'm just going to do penocytosis. This room is a cell. And pretend that I want dust particles. That's what I want. So the membrane, hey, how's it going? Pretend I want some dust particles. So I just kind of like pinch inward like this. So I've got this vesicle, this little bubble here. That's what we call a vesicle, that bubble. If I pinch inward, then I probably get some dust particles. Yeah, there's dust in the hallway, so I probably got some dust particles. Okay, what if I covered myself with like inside-out tape, like reversible tape, and then I went like this for a little bit, and let's say it's kind of like there's wind in the hall. I kind of go like this for a little bit, and then I pinch inward. Won't the tape have like grabbed some of the dust? Like, won't I have some dust stuck to the tape? That's kind of what the receptors are doing. So here's the idea. I've got all these ways of like getting stuff in and out of a cell. Anytime I'm moving in like 1 or 2 or 3 things at a time, I'm super accurate, but super slow. If I need to be faster, I can do something like penocytosis. I can bring in like thousands and thousands of molecules all at once, but it's less accurate. All I wanted were the blue dots. So that's less accurate. This one is medium speed, medium accuracy. So if you think about it, a channel, a gated channel, a pump, something like that, those are, you can sort of classify it like this if you want. They're like really high accuracy, but really low speed. They're super slow. I'm bringing in molecules single file, one at a time. Imagine you guys are going to some big concert and there's only one door, there's one security entrance, and they let people in one at a time. That takes a while, but it's more accurate. Over here, when I look at something like penocytosis, that is very low accuracy, but it's really fast. It's high speed. So sometimes I need that. Sometimes speed matters more than accuracy. RME, receptor-mediated endocytosis, is like medium accuracy, medium speed. It's the compromise. All right, so there's blue dots out there. If I've got to be super picky and accuracy matters the most, this is probably how I'm going to move them in. There's blue dots out there. I need them in here as fast as possible, and if I'm not that accurate, who gives a crap? Cool. Just kind of do some penocytosis, and maybe 10% of what you bring in is what you want. All right, high speed, low accuracy. Or if I'm kind of in the middle, I'm like kind of mid, I want medium accuracy and medium speed, receptor-mediated endocytosis. I can use all of them to bring in blue dots, but in some situations, accuracy matters. In some situations where the blue matters, speed, and in some, it's kind of medium. So up here, this is what it actually looks like. If you're in like a cell biology class, they start getting into like all these different proteins that like join together to like pinch inward. And with RME, we make like this little cage. And so up here, you can kind of see they've got all these kind of cool looking proteins. I think they're cool looking. If you're looking for something to dress up as for Halloween. Clathrin, I mean, come on, that looks awesome. You walk up, they'll be like, what are you? And you just kind of be like, Clathrin. All right, here's kind of a video of what it looks like. And then I'll show you what it looks like under a microscope in real cells. Let me turn down the volumes. Some of you guys, this will explain an interesting disease. Some of you guys have it's a very long and not very original name, hypercholesterolemia. Emia means it's in my bloodstream. What does hyper mean? Yeah, more, above, too much. So hypercholesterolemia means like, hey, my blood cholesterol is too high. Do any of you all, might be your family, or somebody you know, do any of y'all or somebody you know have like high cholesterol just runs in your family? It's like, hey, that guy eats right, he exercises all the time, and somehow he has like really high blood cholesterol. Yeah, so I have naturally high LDL blood cholesterol. So I have blood cholesterol like somebody that was much worse condition than I am. So here's one of the issues. Most of us, we have these receptors, and if there's cholesterol floating around in my bloodstream, my body will pull it out of my bloodstream. If cholesterol is in my bloodstream, it can clog arteries, just like triglycerides can. So I'm supposed to have these little receptors, RME, that are supposed to grab cholesterol and like bring them inside of the cell, and now my blood cholesterol goes lower. So let's say that you are great at processing cholesterol. I give you a piece of bacon. I eat an equally sized, exact same amount of fat piece of bacon. An hour later, your arteries are clean. An hour later, mine are like 50% clean, because my body is not as good at removing cholesterol from my bloodstream. So what I do is I make these receptors for cholesterol. All these other various proteins, some of them are light green, some are darker green, they all kind of join together, and then the cell membrane just sort of like pinches in. And now that cholesterol isn't floating around in my bloodstream out here. However, and mine's pretty minor, there are people that their DNA that codes for this receptor is messed up. So they are horrible at pulling cholesterol out of their bloodstream because these receptors are not quite the right shape. So they're not as grippy. They don't grab cholesterol as well. And so that's when you're looking at some dude and you're like, that guy's super lean. He's like a elite, like a cyclist or something like that. Runs marathons, whatever, and you're like, how does he have high blood cholesterol? You're like, what does he eat? And you're like, he eats like a professional athlete or something. How is that guy? How's this like elite, like athlete, cyclist, like Tour de France, whatever? How do they have high cholesterol? It could be genetic. Your body's just not good at pulling out the cholesterol out of your bloodstream. All right. So then, here's where it gets kind of cool. See how they make Clathrin look like that? That's because Clathrin looks like that. I'll show you. Here's Clathrin. So under a microscope, you're like, it's kind of grainy. It's like, yeah, we're super zoomed in. Keep in mind, this is an individual chain of amino acids that we're looking at here. And here's what's weird, is when those Clathrins join together, like if I have something that looks kind of like a letter Y, kind of like a curvy Y. If I have like a Y and 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 they join, they all kind of make they join up and they basically make like a soccer ball. So this is not computer generated. This is actually like a photograph inside of a cell. So this is, if the room is a cell, I'm in the middle of the cell and I look at the cell membrane as it's doing RME, as it's like pinching inward and bringing in a cage full of stuff. That's the cage full of stuff that it's bringing in. So this is happening in your body right now. How many times? About 2,500 times every minute per cell. And we estimate that you're 80 to 100 trillion cells. And if it doesn't happen properly, I'm probably going to die. So all of this is happening while everything else that we learn about in this class is happening. So here's a cell that's pretty busy importing stuff. Clathrin, clathrin, like every one of those, every like little capital Y is a clathrin, and they link arms and sort of like pinch inward. That's inside of an actual cell. Super weird. I know. I find it kind of fascinating. One thing that's kind of interesting, an interesting application of this, is let's say you do have high blood cholesterol. Then they're like, all right, we can put you on cholesterol medication. So the number one, biggest grossing, greatest selling, most prescribed prescription of all time is Lipitor. Lipitor and then other cholesterol-based drugs. So if my blood cholesterol is too high, I can take a pill and all of a sudden my blood cholesterol drops. You're like, well, why doesn't everybody take that? Here's why. I take the fat from my bloodstream, if I'm using Lipitor, and I store it in my liver. So I end up getting fatty liver. If I get a fatty liver and it's not because of medication, that's because I'm an alcoholic. So I'm basically like storing the extra fat in my liver, the same ways that alcoholics do. So for me, I almost never drink alcohol. So if when I'm old, my blood cholesterol is still really high, I might take like a small dose of Lipitor, and I can start loading up my liver and I'll be okay. But if I'm one of those people that like drinks every week or something like that, then when it's time for me to take Lipitor, my liver's already like kind of messed up, so I might be in trouble. So that's why we don't just prescribe things like Lipitor to everybody. Next picture is a little bit gross, but not too bad. That's a liver. Now your liver is actually supposed to be like this dark brownish, purplish red color, like maroon. But this one is so full of fat that it's not. So the liver itself is still maroon, but all the fat cells in it are yellow. And so the overall color looks yellow from a fatty liver. So anyway, kind of interesting. Disgusting, but interesting. All right. Any questions on these things that are up here so far? Yeah.
Why would you not want it to always be accurate when it's blown out?
Yeah. So you're like, wouldn't I just always want accuracy? It depends. What if I need 1 million blue dots within the next second, or as a cell, I'm going to die? If I let them in one at a time, it's just not fast enough, so I'm going to die. So if I do this, I let them in and I get millions of blue dots, but now I have to like eject the green dots, the black dots, the other ones that I don't want. So it just all depends on like how fast do I need it. You can imagine like my blood sugar right now, shipping blood sugar in and out of my bloodstream is pretty easy. But if I'm sprinting, that gets harder to manage, and all of a sudden like I need sugar like now, like get it in there fast. So it just all depends on the situation. That kind of makes sense? Yeah. All right. And then that brings up the last one, exocytosis. And exocytosis is exactly what it sounds like. If stuff's exiting, if you're just getting rid of stuff. They don't differentiate, are you barfing out liquid or are you barfing out solids or what? They're just like, hey, it's exocytosis, so exit. And if we look at like exocytosis under a microscope, it looks like that. Exocytosis. So there's a cell, and it's just kind of going like Duh. Now, that might be waste material, or it might be like a hormone. So Halloween's coming up in a month, and I turn the corner, somebody freaks me out, and I get an adrenaline rush. It could be that my adrenal glands, they sit on top of my kidneys, just dump a bunch of adrenaline out. How do they get rid of all that adrenaline and dump it into my bloodstream so fast? Those particles could be adrenaline. It could be waste material. It could be that I did this because I wanted the blue dots, and now I exocytose, I barf out all the green dots that I didn't want, or something like that. So it could be anything. Just because it's exocytosis doesn't mean that you're getting rid of a bad thing. Good things leave cells too. Right now, everything over here was passive. Everything over here so far is active. Now we're going to look at some stuff that's not really transferring stuff in or out. So it's not like a type of transport. Here, I'm bringing things in, I'm bringing things out. So one of them that we saw was cell identification. We saw that last class. I have a protein, and there's some carbohydrates hanging off of it. So I call it a glycoprotein, and that's how I know that my blood type is A or B or AB or something like that. So we talked about cell identification. Another one, the default shape for cells is always a perfect sphere. Are all shapes of cells a perfect sphere, though? Nope. So they're not all perfect spheres. So let's say that I've got a perfect sphere right here. And I want to make it look like a red blood cell. What's the shape of a red blood cell look like? Like if I'm looking at it from the side. Yeah, it's kind of more flat. It's kind of like concave at the top, concave in the middle. It's almost like I was pinching dough together to make a donut, but I never quite like had my fingers connect. So a red blood cell from the side looks something like this. All right, cool. So for whatever reason, that helps red blood cells be red blood cells. The theory is they kind of stack in a single file line a little bit better if they're shaped like that. All right, cool. So how do I get a cell to go from being a circle or a sphere in 3D world to looking like that? Well, I can have some other proteins that act as anchors. And so what I can do is I can have, I'll label it like A. There's an anchoring protein. And what I can do is I can put some of these anchoring proteins on the top and bottom of the cell. Then I can take these protein fibers and kind of connect the top and the bottom. And then what if I can shrink those blue protein fibers down? Doesn't it sort of like pull down the ceiling and like pull up the floor? And now it looks like this. Like that. So I can have anchoring proteins that are also in there that help determine the shape of a cell. So imagine, pretend that this room, it's currently like got 9-foot ceilings. Pretend that I wanted this edge of the room to have 9-foot ceilings. Like right there in the middle, I want the room to be like 4 feet tall. So imagine that I wouldn't bust the room if I did this, but I put some bolts in the bottom, some bolts in the ceiling, and then I got steel cables and I connected them. And then I just like tightened down the steel cables. If the room didn't bust, wouldn't I be kind of like pulling the floor up and pulling the ceiling down? That's what I'm doing. So these are called anchor proteins. So I've got like cell ID, glycoproteins, and then these guys act as anchors. You don't have to know it, but one of the most common types of proteins that do that are called ankyrin. So they kind of tighten down like that. And here's what's sort of cool is if I look at, I think I got a picture of it. Yeah, here we go. Here's some red blood cells. I can see they got kind of this cool shape like that. Now imagine this room is a red blood cell. If I looked up at the ceiling, shouldn't I see some of these anchor proteins if they're big enough? And shouldn't I see some of these like blue cables if that's big enough? Like I ought to be able to see it if I'm looking at like the ceiling and the floor. So if you cut open a red blood cell and look at the ceiling, that's what you see under a microscope. Again, these are all real images. It's not like a computer-generated image. It's a microscope image. So I can see that anchor protein there, and there, and there, and there, and there. And then I can see all these kind of like cable proteins that are like holding it all together, like there, and there, and there, and there. It's pretty cool. So anchor proteins. All right. Feeling okay on all of this stuff. Okay, now we're going to get into stuff that you're going to find more interesting. I never tell you if we're getting ready to start something that's like, hey, you're not going to like this. So always wait till it's at the end. So the next thing that we want to do, if you're following along in your notes, now it might be useful to follow along in your notes. Can I borrow yours for 1 second? So far, you're like just focusing on this picture. But now there's a little spot where I can point to something in your notes, and it might be worth it to actually look at it. So here's page 1 on the front. If I flip over to page 2, and I go down to the very bottom right here, it says concept 5.5 right there. That bottom like inch and a half or whatever. 5.5. We're going to be looking at those. So we'll finish off class with these. 5.5 right there. That bottom part says, concept 5.5, a membrane plays a key role in a cell's response to environmental signals. So we're going to look at, thanks Isabel. So we're going to look at like 4 main ways that cells can sort of like communicate with each other. One way that cells can communicate with each other is like if I send a chemical and you receive that chemical, I could send you a chemical called growth hormone, and if you receive it, then you start dividing and growing, things like that. So I'm going to erase this. Does anybody need like this half over here? What's up, Turner?
If you put Benedict in that and cook it and it turns that color, was it? So
See how hot it is?
it's hot. It's been in there.
Nah, that's fine. That looks like Biorette's solution. Biorette's is purple.
No, it's not. It's Benedict. It's Benedict in water and something. I'm not going to tell you what's in it.
I'd rinse it several times because it looks like there's protein and Biorette solution in there. So that's odd.
What if I told you that it's like spit in there?
Then your spit has protein in it.
Is that what?
Biorette's test for protein, but Benedict's.
That's all we've had in the brand new tubes with Benedict.
Oh.
Isn't that weird?
I don't know. I'd have to see you replicate it. Maybe it just means that you're like a falcon through and through, so you spit purple. Okay, so we've got like these few ways that cells can kind of like interact with each other or communicate with each other. So the very first one is called direct contact. Is that number 1? It's called like juxtacrine or direct contact. Yeah. Okay, so I'll go in order. Direct contact. So this is pretty interesting, especially if you're interested in like cardiovascular stuff, like how the heart works. So number 1, we'll call this like cell communication or cell signaling. How do cells kind of communicate with each other? One is direct contact. All right, cool. So your heart cells. They're never going to ask you exactly how do heart cells work on the AP Bio test, but this gives you an example of how direct contact works. So your heart's kind of interesting because, let me grab a picture of a heart. I don't know how many of y'all have ever seen like a human heart. Have any of y'all ever like toured an anatomy lab and you've like held a human heart? Couple? UT Southwestern in Dallas does that. You can go on like tours and stuff. Let's see. I hadn't planned on showing you a picture of a heart, so give me just a second. I gotta grab it. All right. So here's one that's preserved. Whenever you preserve something to dissect it, you suck out all the blood because the blood will rot, and you don't want to dissect like a rotting animal. That's disgusting. So we suck out all the blood, and then we replace it with preservative. And so that's why preserved animals smell like preserved animals. If you were blindfolded and you walked into my Bio 1 class at the end of the year, whenever we're dissecting rats, if you didn't know that they were rats, you would say, and I was like, hey, what do you notice about the smell? You would say, like, it smells like somebody used a bunch of cleaners in here. Like, people were, like, cleaning the counters, the windows. It doesn't quite smell like Windex. It doesn't quite smell like 409, but it smells like, I don't know, some weird chemicals, like I'm in a pool or something like that. So that's what it looks like if it's preserved. Here's what one that's fresh but clean looks like. So it's got a little bit more of the original colors. So that's a heart. Well, a heart's interesting because it needs to beat from the bottom up. So I beat from the top down to fill up the ventricles with blood, but then to like pump blood out to my body, I need to like squeeze from the bottom up. If the heart just goes like this, I don't get it all. Imagine I've got a tube of toothpaste. So here's a tube of toothpaste. And I just grab it in the middle and I go like this. Some toothpaste came out the top if the lid is open, but didn't like half the toothpaste just get sort of like shoved down to the bottom? So if I want to more efficiently get toothpaste out of the tube, don't I need to like squeeze the tube from like the bottom up and now I can get like most of it? That's what's happening down here with your heart. So I have to somehow get it to contract, like the bottom part contracts first and then like this, so it's kind of like squeezing from the bottom up. So you might wonder, like, how does that happen? It happens because the cells are joined with a type of junction called direct contact. So, and it's also called juxtacrine, but that name's dumb. So they call it direct contact is correct. You can call it that, and that's the official name, but you can also call it juxtacrine. So it works like this. Here's a cell, and it's got this channel. So it's got a protein channel, just like a moment ago before I erased this, I had a protein channel like right in here. It's got a protein channel. Cool. Here's another cell. Man, need to get like real erasers. Here's another one. It's got a protein channel. But what I'm going to do is I'm going to line up that channel on that cell with this channel from this cell. So there's one, and there's one, there's one, and there's one. Over here, I've got a channel. And just so you can kind of see it, I'm going to make this one green. This one has a channel that just lines up with that one. So it functions as if they've got a hallway between them. But basically, this cell is providing like half the hallway, and that cell is providing like half the hallway. All right, now, the next part is beyond the AP Bio test. You want to be able to think like this, but they're never going to ask you, like, how exactly does the heart beat? Your heart's kind of weird because it will beat on its own even if it's not attached to a brain. So your heart obviously needs oxygen. It's obviously got to get rid of CO2. You got to feed it sugar, otherwise it dies. But if we cut all the nerves to your heart and we're still feeding it sugar and feeding it oxygen and getting rid of the CO2, your heart will just keep beating on its own. It's called automaticity. It just beats on its own. My legs don't do that. Like, my leg muscles move when my brain tells it to. And if I snap my spinal cord, like, I'm paralyzed. Like I will never move my legs again. I snap my spinal cord. But if I damage the nerves to my heart can still beat. It might not beat the right speed. It might not beat perfectly, but it'll still try to beat. There's a famous scene in an Indiana Jones movie where the guy gets his like heart ripped out, and it's like beating in front of everybody. Have any of y'all seen? We watch totally different. Okay, so a few have. They're doing some sort of like weird like voodoo ritual or whatever. So there's some slight truth in that. If I take the heart out, it could still beat. So here is how your heart beats. I've got calcium. I've got a calcium leak channel. I'll make it a different color. It'll be purple. Calcium leaks in there. All right, cool. Turns out, if 100 calciums, I'm just picking a number 100. I have no idea how many calciums. I'm sure it's billions. But just for easy math, if 100 calciums leak in there, then that cell contracts. Okay, same thing with this guy. If 100 calciums leak in there, that cell will contract. Same thing with this guy. If 100 calciums leak in there, that guy will contract. Have you guys ever heard that your heart has a pacemaker in it? There's some cells that set the pace. I don't know, it's forensics. Maybe somebody's being murdered. I'm not real sure. Yeah, I'm not sure. So imagine that we're all heart cells. We're all joined with direct contact. We've got these gaps in here. This type of direct contact is called a gap junction. There's a few different types of direct contact. This is called a gap junction. Kind of makes sense. There's like a little gap in there. So even though these are three separate cells, on the inside, if calcium leaks into this first one, can't the calcium like move throughout the second and third cell and back and forth and everything? Almost like if you ever been in a hotel and it's one of those party rooms, and so like on my hotel wall there's like a door, and I can actually like join it up if I'm buddies with the person next door. I can like open up the door and you're like sharing that. That's like a gap junction. So next time you're at a hotel and somebody's like, what's that? And you're like, oh, it's like a gap junction in a heart. That's what it is. So I've got this gap junction. Okay. So here's how this works. There's a spot in your heart right about here. That's where electricity is supposed to start. This guy is supposed to fire, send electricity down to the bottom, and then that electricity dissipates bottom up, and your heart contracts perfectly. If your pacemaker cells are in the right spot, everything's great. If it's in the wrong spot, you got problems. So here's how it works. Pretend that we are all heart cells. We are all joined by these gap junctions. All of us are going to start leaking in calcium. So there you go. This guy's going to leak in calcium. This guy's going to leak in calcium. What happens if you get to 100 calciums? If you're a heart muscle cell, what happens when you get to 100 calciums? You contract. Okay. So I need to somehow have this happen, but have it be like a coordinated motion. I don't want random contraction. All right, so we are all heart cells, but we all leak calcium at different rates. So some of us are leakier than others. All right, so let's say that you guys, kind of in the back corner of the room, you guys leak in calciums like 50 per second. You guys in the middle of the room, more like 60 per second, 70 per second, 80 per second, 90 per second, and I'm the leakiest. I leak in 100 calciums per second. I always make fun of myself over you guys. So I'm right here. So here we go. The heart contracts. Whenever it contracts, all the calcium exits. So all the calcium exits. You guys leak in 50 per second, 60, 70, 80, 90. I'm 100. So we now have a race. 1, 2, 3, go. After 1 second, who leaks in 100 calciums first? Me. So I contract. But because I contract, and we're sharing the same internal space, then you guys contract. But I contract first, so then you guys contract second, you guys contract third, you guys contract last. So isn't there like a wave of muscle contraction going across the class? That's how your heart beats. Now, we all just contracted, so everybody just ejected all of their calcium. Now we're going to have another race. We start leaking in calcium. Who leaks in calcium first? Me. So I set the pace, so I'm called the pacemaker. And if I'm in the right spot, your heartbeat is correct. However, what if the leakiest cells happen to be right here? Let's say that I'm still the leakiest cell, but I'm located right here. Now doesn't my heart beat from here outwards? And so that's why I'm going to die. So what they'll do is they'll go in and they'll install a pacemaker. So we can get a synthetic device. and we install it like right under your collarbone, and it sets the pace for you. So now we have this little bitty system, and about once a second, that machine shocks your heart so that it has the right pace. Otherwise, if we shock your heart and it contracts from the middle outwards, your heart is beating incorrectly. Again, that's kind of like I'm supposed to squeeze the tube from the bottom up. If I squeeze it from the top down, like that's not going to work. Yeah. A heart arrhythmia is any time you have an irregular rhythm. It could be because my pacemaker cells are in the wrong spot, but it could be something else. That's kind of like if I say I'm talking about dead batteries and that's why cars don't start. If you're like, oh, is this why cars don't start? It's a reason that a car might not start, but there's other reasons too. So if I have a bad enough heart arrhythmia, if my pathway of contraction is wrong, they'll install a pacemaker. So what they'll do is they'll actually burn out the bad pathway. So, the first thing they'll do is this. Right now, the heart's beating and the leakiest cells are right here, so I contract first. So what they'll go in is they'll burn me to death. So I die. Okay, now, what they hope is that the second fastest pacers were back where it was supposed to be. And if it is, cool. We killed 0.0001% of your heart tissue, so you'll be fine. And now, everything's working like it's supposed to. But if instead, the second fastest pace is right here, and that's still really bad, then it's like, all right, well, maybe we like burn out that second spot. And then a third spot pops up. So at some point, we're like, all right, we're going to have a machine set the pace for you. So you install this right below your collarbone, like this lady. There's a little pocket, kind of like a shirt pocket, and you just put a pacemaker in there. Got a pacemaker in here. Unless it's in the back. Oh, there you go. So, old original version. They just cut a little pocket. They cut through the skin, cut through the muscle, and they make a little pocket, and they just like slide it in there and just sew it back up. Batteries used to last about 10 years or so. So if you're 90, that'll last the rest of your life. If you're 50, then about every decade, they cut open the pocket and they can change out the batteries or put in a new device. So if it's been a decade with pacemakers, technology is kind of like cell phones. So they generally don't change out just the batteries. It'll be like, hey, you know, we've got the iPhone 17 now. So we've got this pacemaker now. So instead of this one, we put in this one. This one's more similar to what was on the screen earlier. This guy. And then I'll have these wires. If you look at an x-ray, I've got a pacemaker that sits right here, and then these wires just go in, and they're just shocking my heart. Pacemakers are smart, so they can actually see, they can check your heart rhythm, see how often it's beating, and they'll only shock your heart when you need it to. So now I don't waste battery. So it might be like, hey, right now my heart is pacing on its own just fine. So this pacemaker is just sitting there like, everything's great, everything's great, everything's great. He missed a beat, shock it. And it'll shock. Take your little phone, download information, you put this little thing on your chest. It might be more advanced nowadays. Take this thing on your chest, download the information. It'll tell you how much battery life you have left. It'll tell you how often you're pacing and stuff like that. My wife has a pacemaker. She has this disease called Wolff-Parkinson-White syndrome. And so in her heart, the pathway that electricity travels was wrong. And so her heart, she'd just be sitting there. And her heart rate would sometimes go up to like 220, 230. 180 beats a minute is 3 beats per second. So I don't know if that's 3 beats a second or not, but I know I have to go every second. So imagine your heart is beating like that. That's like a hummingbird or something. Only you're a human and you're sitting there doing nothing. So it feels like your heart's going to explode. So what they went in is they went in and they're like, all right, you got this bad pathway. We're going to burn it out, and then hopefully the good pathway takes over. Hopefully that was the second fastest pace. And they went to burn it out, but before they went in, they're like, hey, your bad pathway that we have to destroy and your good pathway are right next to each other. So we might destroy the good pathway also. Accidentally destroyed, to some degree, the good pathway also. So she's got a pacemaker. She's had it for, she's on her second one now. She's had it for 14, 15 years. So she's the youngest person that walks in there most of the time. You go in there with all the people at the pacemaker place, everybody's like 70 or 80. She's like, she doesn't walk like that. She's like, does your wife walk like that? So yeah, pacemakers, it's kind of interesting. I know, I didn't know if you were going to say anything about that or not. Since you volunteered, do you, what disease, like why do you? Oh, okay. So you have a vagal nerve, so syncope is like out of rhythm, right?
Yeah.
So what exactly is vagal syncope, do you know? Or like what causes it?
I constantly forget.
Okay, cool. Do you know if it's inherited or is it just like a random chance?
I think it's inherited. My mom has like something similar, but I got the worst of it.
Got kind of the worst of it. Okay.
Almost died in kindergarten. Great.
So how long have you, so did you get a pacemaker in kindergarten?
I did. I got a new one last November.
Okay. And they gave you like a whole new pacemaker, right? They didn't switch out the batteries?
But, yeah.
Cool.
I got two of my old one. It's in a dorm now.
That's cool. Like these, in case you were wondering, you're like, hey, Cotton, have you just got like pacemakers sitting in there? Like, aren't those expensive? These are just like models. So if I go in kindergarten and they're like, we're going to have to, install a pacemaker, they'll be like, it's going to look like this. So this just has like a lump of metal in it. It's just the outside of a pacemaker. It's not like I've got some sort of like $50,000 piece of equipment and I'm just like, or something like that. So these are just like the models in the doctor's office when they're going to be like, we're going to put this right under your collarbone. So you can see it in most people because it is pretty shallow. This is flip-flopped. So the doctors weren't wrong. They know that the heart is on the left side. That's actually under her left collarbone instead of right. It's just doing the flip-flop thing. But like my wife's, if she's wearing a shirt like this, you couldn't tell. If she was wearing a T-shirt and it was tight, you could tell. If she's wearing a bathing suit, you can be like, there's a little, like I can see something that looks square right there. She's also lean. So again, if I was like heavy enough or it was buried deep enough, you might not be able to see it as much, but like you can see it for sure. So kind of interesting. So direct contact, gap junctions. Who would have thought?
What if it gets cut?
Do what?
Like what happens if she gets a cut on the top part?
It's all scar tissue. So like scar tissue generally grows around, anytime you have a void in a place that you're not supposed to have a void, like obviously like I'm supposed to have chambers in my heart, I'm supposed to have airways in my respiratory system. But anytime you have a void in a place that you're not supposed to, your body usually either fills it with cells or fills it with scar tissue. So even if you were to like cut open that exact cut, there's probably like scar tissue kind of like gripping to it that you have to like take it out. I don't know that for sure, but when I originally make the cut, I slide it in, but then like scar tissue will kind of grow and like fasten it down. But yeah, she can't go through metal detectors and stuff like that anymore, which who cares? All right. Yeah. Okay, so direct contact, gap junctions. The second one down is the next one, endocrine or exocrine? Number 2, paracrine. Oh, that's okay. Number 2 is paracrine. Paracrine signaling. And then that mean is 3 endocrine and 4th is synaptic? Third is synaptic and 4th is endocrine. Okay. So I'll go in the same order that you guys have it. So this is a local signal, a close by signal. So paracrine is when we're all skin cells, and let's say that you guys have likely heard of like the endocrine system maybe, but you might not have ever heard paracrine. So paracrine and endocrine are similar, but not identical. They're different. So paracrine signaling is if I'm going to send out a local signal, a local chemical signal, and only the people nearby should react. It's local. It's not body-wide. Hormones, that's the endocrine system. We'll talk about those more in a second. Those are body-wide. When I dump adrenaline into my bloodstream, like The adrenaline comes from the top of my kidneys, my adrenal glands, makes it all the way up to my brain, and then like my whole body responds. My eyes dilate, my heart rate changes, respiratory changes. That's like a body-wide thing. Sort of like right now, I'm teaching in this room. This is like local information. Only people in this room understand what's going on. Down in the physics hall, they have no idea. So this is kind of like paracrine signaling. Endocrine signaling would be like if the principal gets on the intercom and makes an announcement for the whole school. It's school-wide, as opposed to just like right here within like the radius of my voice. That's more like paracrine. So here's what will happen. Let's say I've got a bunch of skin cells. Doesn't matter how many, I'm just drawing a bunch of random ones up here. This is pretty cool. It explains all kinds of things that are great and fascinating when they work well, and it also explains some stuff that's a disease process that's not working so well. Let's say these are all skin cells right here, and I get a cut. So right there, I get a cut, and these skin cells die. So they're all gone. All these guys are right here in the middle are gone. Do I want to just leave this open wound day after day after day after day? No. So what I want to do is have it grow back together. Right now, I'm 49, and yeah, there's some deep cuts where I've got some scars on me, but can't you imagine that on my forearms, I've probably gotten teeny tiny little cuts on my forearms thousands of times? I've moved from this house to that house, moving boxes. I've had to, like collect wood for the fire, cut down a tree, whatever. So you can imagine I've had like thousands of teeny tiny little scratches on my forearms, and like you can't see anything right now. So where do the new cells come from? I had like, let's say 10 of them died. Where do all the new 10 replacements, where do they come from? Yeah, the other skin cells, so the nearby ones. So basically that guy has to somehow realize his buddy is gone. He needs to divide and then fill the gap. This guy needs to realize his buddy is gone. So he needs to somehow know to divide and fill the gap. So I need to somehow sense when I'm shoulder to shoulder with other skin cells. And anytime I'm not, I'm supposed to just grow and fill the gap. That's how we heal a cut. So let me give you an example. Let's say all of us are skin cells. So there's like 32, 33 counting me, if everybody's here. So 33 of us are skin cells. And all y'all, you just died. So I just scratched you away. Sorry. You're irreplaceable, but we're going to replace you. So you like 8 or 9 are dead, like all 10 of these seats are empty. Okay, cool. So who should fill those seats? You guys. You guys on this edge should divide. And so now for every one of you guys, you double and you'll fill those seats. You guys over here, you should somehow sense that there's a void, divide, fill those seats. Okay, so right now, there's a gap in this seat. So shouldn't somebody around it, behind, in front, side or side, shouldn't somebody divide and fill that gap? Yes. How do they know there's a gap, or how do they know when they're done growing? Because here's the problem. Let's say that 10,000 cells die, and I replace it with 10 billion. What's that called? Cancer. Anytime cells grow when they're not supposed to, that's cancer. So cells have to know when to grow to like fill the void, but they have to know when to stop, otherwise it's cancer. Cancer always ends in tumors because a tumor is just this extra lump of cells because cells are growing out of control. All right, cool. So here's how it works. It's paracrine signaling. Have you guys ever read or seen the video, Horton Hears a Who? You know how at the end, they're like, we are here, we are here. That's Horton Hears a Who, right? They're like, we are here, we are here. And they're like trying to listen for it. Your cells are always sending out chemical signals, paracrine signals locally to the guys next to them that's saying, we are here. So I am producing this chemical that hits a receptor on you, and that chemical means we are here. So think of it like this. Let's say that we're all cells. I'm a cell, and I'm sitting right here. All right, so the way that Ian knows I'm here is because I say it very, very quietly. We are here. Okay, can you hear me? All right. Can you guys way over there, can you hear me? No, but I'm talking. You can see my mouth moving like, I am here. I am here. I am here. So I'm saying I am here. The way that a cell is doing is I'm sending out a chemical. He has a receptor that's receiving it, so he knows that I am here. Now I die. It's gone. Is he getting a signal from his right anymore that says I am here? No. So what does that mean? That means there's a missing cell. So what is he going to do? He's going to clone himself. He's going to grow and divide, the whole mitosis thing, and then like fill the gap. How will he know that we've filled the gap? Because once he's cloned himself, there'll now be somebody sitting here going, I am here. So I'll start sending out the chemical signal that says I am here, and he knows to quit growing. However, what if, I got you. You're welcome. What if this cell over here, that receptor is broken? Do I ever get the message of we are here? No, so it just starts growing out of control, and that's cancer. What if he doesn't send a good copy of this? Then I never receive the message, and I will grow out of control. That's another version of cancer. So we study cancer more in Unit 4, but it's interesting. Paracrine are local signals nearby. If I get a cut right here, and it gets inflamed and infected, I'll know it because it's all red and puffy and maybe even hot to the touch right here. That's a local reaction. When I get an infected cut right here, my left shoulder doesn't turn red and puffy and inflamed. That's body-wide. This is just a local thing, paracrine signaling. So it's how cells talk to each other. To give you an idea of how it works, we got a video that obviously starts out as an animation first, but then they switch over to a microscope view of real cells. So they take some cells, some human skin cells, and they just smear them on like a glass plate, like a microscope slide. And they're all like side by side by side like this. Then they take a needle, and they just like scratch right down the middle, kind of like when I killed like these 2 rows of skin cells. Okay, so now these are real cells. There's a gap, and so the cells just keep on growing. They grow and grow and grow. I'm listening. I'm not getting the we are here chemical paracrine signal. I'm still not getting it. I'm still not getting it. I'm still not getting it. And then eventually, I bump into the guys across the way, and that's how I know I'm done. And that's how my skin knows to heal. It's pretty cool. What I like about biology is they always have these like really simple solutions to something that seems like it should be super complicated. Like, how does this skin cell, it doesn't have a brain, how does it know when to grow and when not to? And all of a sudden it's like, well, this sends out a chemical signal, and if it's absent, you grow. And if it's present, you don't. And you're like, oh, that's actually a pretty simple solution to something that should be super complicated. Here's another example. Bacteria. This is a white blood cell. These are red blood cells. This is under a microscope. And this guy's a bacteria. He's sending out, accidentally though, a chemical scent. And this white blood cell is following that chemical trail. So he is following the local scent. Kind of like, again, it's popcorn day. It's supposed to be popcorn day. I thought there'd be like a wafting of popcorn smell, and if I was blindfolded, I could probably like trace the smell down to where it started. So this is a white blood cell that's following the chemical trail. The neutrophil is the white blood cell that's following the chemical trail of this bacteria. Oh, I'm sorry, it's that bacteria, not this one that he's chasing. That one's something stuck to him. So he's following the local chemical trail, the chemical scent, kind of like when somebody puts on like super strong amounts of body spray and then they walk past you in the hallway or whatever. Like that. Your life depends on this guy. And then eventually, phagocytosis. Yay. One down, 100 billion to go. All right. The next one. It's called synaptic signaling. Synaptic signaling. Synaptic signaling, you know how like osmosis is just sort of like a specialized version of diffusion, like if it's water diffusion, we happen to call it osmosis. That's what synaptic signaling is. It's basically paracrine, but it's only neurons, nerve cells talking to neurons, nerve cells talking to nerve cells, neurons talking to neurons. So it's like just paracrine signaling, however, it's just nerve cells talking to nerve cells. And you guys have heard of this word before. What are the chemicals that nerve cells use to talk to each other? It's kind of a long word. Starts with an N. Helps them transmit messages between neurons, neurotransmitters. You might not know that word. So, in my brain, I've got 80 to 100 billion nerve cells. Pretend that each one of these black lines is a nerve cell. And again, in reality, I'd have tens of billions of them. Some estimates say as high as 100 billion, some say 80 billion. It all depends on your height. The taller you are, the bigger your brain, the bigger your hands, the bigger your liver, everything. So if I'm 7 feet tall, I have a bigger brain than somebody that's 6 feet tall. So if you ever hear that males on average have larger brains than females, that's true, but that's only because males are on average taller. So a 6 foot tall lady and a 6 foot tall guy should have the same size brain. So it's not really your male or female, it's more like how tall are you? All right, so neurons communicate with these chemical signals. They talk to each other with neurotransmitters. So here's what's cool about the brain, and we talk more about the brain later on in the year. Like right now, how many of y'all learning that paracrine is a local signal? How many of y'all that was pretty much new information? Okay, cool. So let's say that I see you guys on Friday, and I come back, and I say, what was the second type of cell signaling? And you're like, I remember that. It's paracrine signaling, and that means it's a local signal. We are here. Here's what you did. In your brain, if nerve cell number 1, 2, 3, 4, 5, and 6 fire in a certain order, that's where you're storing paracrine means local signal. If 1, 2, 3, 4, 5, 6, 7 fire in that order, that's where you're remembering that like heartbeats are related to direct contact. So on Friday, when I see you again, and I go, what does paracrine signal? What does it mean if I say paracrine signaling? Some of you guys are going to go, and then this pathway is going to fire, and you're going to go like, oh, it's a local chemical signal. That's called remembering. Some of you guys, though, I'm going to go, what's paracrine? And you're going to go, I can't find the pathway. And we call that forgetting. So if these guys, if nerve cell number 1 sends a neurotransmitter, a chemical message to nerve cell number 2, who talks to 3, to 4, to 5, to 6, that's where we store information. If I'm doing this other pathway, that's where I stored some other piece of information. So at one point, I took U.S. history, and I knew stuff. Now, I can summarize everything I know about U.S. history on this Post-it note, if I write big. I overwrote all of that with useful stuff, like Golgi apparatus, synaptic signaling, paracrine signaling, things like that. So, synaptic signaling is how nerve cells talk to each other. That's what's happening here. Here's one that's kind of stained. So this yellow thing up here is a nerve cell, a neuron. This yellow thing down here is a nerve cell, a neuron. And this right here is this gap where they almost touch. You don't have to know the name, but it's called a synapse. That's why we call this synaptic signaling. Your teacher might call it a synaptic cleft. And these are neurotransmitters, the purple stuff. So whenever I'm thinking a thought, this nerve cell sends a chemical message to the neighboring nerve cell, who can then send it to the next guy, to the next guy, to the next guy, to the next guy, and that's a thought. T-H-O-U-G-H-T. So this is a mouse brain. It's sliced up and under a microscope. So this is what fear looks like. Right? His last thought was probably like, and now his brain's under a microscope. So you're like, what is that? And you're like, that is fear. Isn't that strange? Maybe not. I'm sure he was happy and he didn't know it was coming. Maybe they approached him from behind. All right, that's it for today.